Normal eukaryotic cells have only a limited capacity of cell division. The process that limits cell division has been termed as replicative senescence. It appears to be a fundamental feature of somatic cells, with the exception of most tumorous cells and possibly certain stem cells, as shown in the diagram. Here we are going to discuss a little bit about the telomere. A telomere is a region of repetitive sequences at each end of chromosome. Telomeres protect the end of the chromosome from DNA damage or from fusion with the neighboring chromosomes. Telomeres serve a dual role in protecting the chromosome ends and in intracellular signaling for regulating cell proliferation. The ends of linear chromosome pose a unique problem. During DNA replication, DNA at every end of the chromosome cannot be fully copied in each round of replication, resulting in gradual shortening of chromosomes. Because DNA polymerase can only elongate from 3' hydroxyl group. Within each fork, one DNA strand called a leading strand is replicated continuously in the same direction as moving fork, while the other lagging strand is replicated in the opposite direction. The replication machinery builds the lagging strand by a backstitching mechanism. RNA primer provides 3' hydroxyl groups at regular intervals along the lagging strand, here shown in the diagram. Whereas the leading strand continuously elongates in the 5' 3' direction of all the way to the end of template. Final primer would provide 3' hydroxyl group to synthesize DNA. Here shown in the diagram, short stretches of adjacent DNA fragments called Okazaki fragments, but the primers would later be removed. As shown in the below diagram, the primers are removed now. The 3' hydroxyl groups are adjacent DNA fragments called Okazaki fragments. The responsibility for sealing this neck lies with the enzyme ligase. However, at the end of the chromosome, there is no hydroxyl group available to prime the DNA synthesis. Because the final primer was removed at the end, because of this inability to replicate, the ends of chromosomes would progressively shorten during each replication cycle and would cause the end replication problem, which finally leads to replicative senescence.